Well, lately I've been thinking about these jumping RNAs and very curious about what natural role they're playing in our during the development of our germ cells, and even during the development of an embryo. And there are ways to knock things out now, knock out whole genes, knock out pieces of genes, in fact, that are very precise. Technically, they can do very precise knockouts now. And I would love to see what would happen if you knocked out, say, all of a certain kind of transposable element. For example, an LTR. Long terminal repeat, which is basically that promoter and enhancer region that I was talking about before that viruses carry around with them, and it's also what transposable elements. So these things, these little fragments of RNA that can jump, they just can't jump outside your nucleus like a virus can. But these guys jump in the nucleus, and they have observed that there's lots and lots of this jumping seems to be taking place during very specific times during the development of a germ cell. And the general idea is it's very destructive, that this is disrupting all kinds of normal, healthy genes, and if we could just turn it off completely, everything would be much better. And everything I've learned about evolution and about RNA and about how genes work, but mostly about evolution, sort of tells you that it's a process that wouldn't just be happening random helter-skelter like a bunch of hooligans taking off, that just needs to be turned off instantly. My gut feeling is that there is really something important happening there, that either the RNA that's being made, say, from those long terminal repeats, they are forming some kind of secondary structure perhaps, that signaling, that some form of tremendously important signaling is taking place at that moment. And again, my off-the-cuff thoughts would be that it has something to do with rebooting a basic germ cell from being a highly evolved cell to being very basic dividing cell, something close to our very most primordial basic dividing cell. Quite different, of course. It's a beautifully evolved, complex cell, obviously. Something that's going to turn into a sperm or an egg is a very developed, evolutionarily developed cell. But there's a moment when it needs to turn off all the networks and all the genes that are not necessary if you're just going to be a basic dividing cell. And my instinct is it has something to do with that turning on all of these transposable elements and long-term terminal repeats and even allowing jumps to take place is somehow rebooting that cell. Once it's turned off, whatever the process is tremendously important to keeping a very basic identity of a basic cell.
So how could that translate into a research or an experiment?
Oh, right. So, it would translate into an experiment by seeing what would happen if you just got rid of them. What would happen to the cell? The dogma predicts that nothing would happen. That, in fact, the cell might be healthier than it has ever been.
If you get rid of all LTR.
Just get rid of all that. It's called junk. It's called junk. Foreigner, parasitic junk. And my feeling is it's doing something. And the cell would not develop normally into a normal egg or sperm if you didn't have that sort of reboot event. So, yeah, that's what I'd do. I would get rid of them all. See what happens.
And your prediction?
I think you would...
Specific prediction.
A prediction would be you would not be able... That cell would not develop normally into a normal egg or a normal sperm, and it may very well become cancers because you've lost some very basic controls over just plain old cell division at that point. So.
Great. Okay, another question. What's important for people to know or be thinking about in terms of RNA's role in evolution, especially perhaps at the very beginning of the history of life?
Oh. Yeah, I think what people might find interesting, or what I find interesting as I think about RNA and its role in evolution, is the possibility that it really was the first replicating molecule. That it was the first thing that was able to survive in that primordial soup and actually replicate itself. And that gave it such an advantage over other molecules that could form, but they would fall apart and couldn't be formed again easily. And then there evolved somehow a sequence of these nucleic acids that were actually able to turn around and remake themselves. And just starting from that point, you can begin to piece together a little bit. There's still huge gaps, like how did that first protein get made? Because the RNA itself is helped by proteins. Proteins are like machines that come in and do stuff for you, and they're very important in getting the ball rolling. How they first came about, who knows? But you know that it has to involve a piece of RNA, and you know that one of the first parts of making a protein is done by something called a ribosomal RNA. So you have RNA that gets read by other fragments of RNA. Then it can make more of itself. So if you start there and you start thinking about the proteins that are needed in order to do that, you would have the ribosomal proteins, but then you would also need, it's nice to have an RNA polymerase making more RNA. You begin to make proteins that help the RNA make more of itself. So it's like the whole system starts to grow based on how to just make more RNA. And that there's something very cool about that. And you can build a whole story starting there.
So does that imply that there was kind of an age of RNA, the way there was an age of gigantism and all that sort of thing?
An age of dinosaurs. Yeah. Absolutely. Yeah, I'm quite sure. In fact, they talk about it. It's the RNA world. It was following the primordial soup, which was just fragments of molecules that were forming because of shots of lightning and stuff like that into pools of warm water, and possibly, of course, it's theoretical. There's no fossils that we can get hold of at the moment, but that the very first early replicating molecules were RNA, not DNA. DNA is a very nice double helix. It's sort of the perfect structure for keeping that information safe and sound and still accessible, but it probably came second.
Is that the inverse of how people tend to think of it?
Yeah. No, the mantra that you learn in high school is DNA gets turned into RNA, gets turned into proteins. And that is certainly true now. But it's a much more intricate story on how it really happened. You have to think of these things as happening. These are little teeny chemical reactions. They are not happening because we're thinking about it. They're just happening. Something, and it took a huge amount of time, happened just because things were bumping into each other. It's extraordinary to me. It's chemistry is pretty cool.
So this is a sort of oddball question, but do you think, despite the fact of complexity that we see today, is that same exact process still happening on Earth?
Right. Is there evolution possibly taking place at that level still?
Yeah.
It may, but what happened was what started being able to replicate itself was so great that it actually sort of took over. It just was so efficient and so much better than anything else that was going on, and still really that's going on now, that it's kind of swooped up the whole story. So maybe there are events absolutely taking place nowadays where you can make a nucleic acid from lightning, but evolution is enough to have created a system that is so efficient that it has not needed to recreate itself again.
Mm-hmm.
What you see now and what you carry in your cells now is, it's something that formed a long time ago and worked great.
Mm-hmm. Yeah, at the expense even of its earliest incarnations.
Yeah. It just wanted to make more of itself, and it just started doing it better and better and better and better. In fact, all we are right now are, a particularly terrific way of passing on our little pieces of RNA.
Packaging
And sending them off to our kids.
Yeah.
Yep.